Hello everyone. Today we are going to discuss about the last topic of fertilization and post-fertilizational changes. Here we have to see what is the process of fertilization in angiosperm. In angiosperm, this process of fertilization is called as double fertilization because fertilization takes place two times. As in last class, I discussed that two male gametes are produced. Here we can see that we will be having two male gametes over here. So these two male gametes are fertilizing two different things. Here we have to see first what is the structure of in the male part of the plant which contains male gamete, that is pollen grain. So this pollen grain is germinating at the stigma after pollination. There are two conditions. If pollen grain will be of correct type or compatible type, it will germinate. If it will be of wrong type, it will be eliminated. Wrong type means suppose the pollen grains of mustard cannot be able to germinate on the stigma of China rose. So that will be rejected. And if it is belonging to the same species, it will be germinated. After germination, the second step is movement of pollen tube in their style. So first we have discussed about stigma. Second step is movement of this pollen tube in their style. In last class I discussed about how pollen tube is formed from the tube nucleus and intine. Intine is the inner membrane of the pollen grain which will be extending out from the germ pore and will be producing an elongated structure which is called as pollen tube. Now this tube is growing down with the help of a nucleus, which is tube nucleus, followed by two male gametes. These are the two male gametes which we are getting in the three-celled stage of pollen grain. So sometimes pollination occurs at two-celled stage, sometimes it occurs at three-celled stage. After pollination, at stigma, selection and rejection has taken place, and then pollen tube has reached to the site of fertilization, which is called as embryo sac. Before entering to the embryo sac, this pollen tube has to enter in the ovule. So for entry of ovule, there are three options. First, it may enter through the chalaza. This portion is called as chalaza. So if pollen tube is entering through chalaza, it is chalazogamy, which is not given in your book, but you will study that in class 12. If it is going through the integument, this is the second point of entry, which is called as mesogamy. But the most prominent way of entry of pollen tube, as you can see over here, this spore-like structure is called as micropyle, and entry of pollen tube through micropyle is called as sporogamy. So in this way, pollen tube entered in the ovule. After ovule, it has to enter to the final destination. What is that? That is embryo sac. So in embryo sac, you have to see how pollen tube is entering. If you remember the diagram of embryo sac, I discussed in the previous class that it consists of synergid, and that synergid is basically having filiform apparatus. So filiform apparatus is actually supporting entry of pollen tube inside embryo sac. So what is filiform apparatus? You can see this is the structure which is called as synergid on either side of the axle. It is having some finger-like projection which is called as filiform apparatus. So with the help of this filiform apparatus, this pollen tube is ultimately opening. This tip is dissolved due to help of some enzymes, and then these two male gametes will be released inside the embryo sac. Now in embryo sac, we have to see focus basically on two major structures. One which is called as this central cell with two polar nuclei, and second structure is in the center at the base, which is egg cell. Now there are two male gametes released. I'm taking another color for the male gametes. Let's say I'm taking green color. So now after entry of these two male gametes, first and second, the first male gamete fuses with the egg cell and will be producing zygote. And this process where the first male gamete fuses with the egg cell and will be producing the zygote. So male gamete is haploid, egg cell is haploid, so zygote will be diploid. So in this way we are getting diploid zygote, and this process is called as true fertilization or syngamy. After this second male gamete fuses with these two polar nuclei. But here the process is slightly different. This haploid male gamete, second male gamete, fuses with the secondary nucleus. These two dotted structure which is present in the central cell will be ultimately fused together and form a single nucleus, and that single nucleus is called as secondary nucleus, and this secondary nucleus is diploid in nature. When these two nuclei will fuse together, it will form a secondary nucleus. So instead of polar nuclei, now it will be termed as what? It will be termed as secondary nucleus, which is already diploid because of the fusion product of two polar nuclei. So this secondary nucleus when fuses with second male gamete will be giving you a triploid structure which is called as PEN, primary endospermic nucleus, which is having three axis structure, and it will be developed into endosperm. So endosperm is providing nutrition to the developing embryo. So this way we are almost towards the end of the chapter, that how fertilization had occurred in case of angiosperm, where the fertilization is taking place twice, and the process is called as double fertilization. After fertilization, as most of the things we have seen, that the ovule is converted to seed, and ovary is converted to fruit. These things are called as post-fertilizational changes, which is given here. You can see the first step is, this is called as post-fertilizational changes. When we talk about these changes, the first step is the things which are withered off or fall off, that is petal, sepal, stamens, style and stigma. These are the things which will dry up and fall off. Most of the cases, stamen also, sepals also withered off. But in case of brinjal, if you have seen that bangan, you call them in Hindi, this structure in the brinjal is basically the persistent calyx, which is becoming leathery, thick, persistent, and attached at the surface of the ovary. So sometimes this calyx is persistent, as in case of brinjal. Now the most important thing you have to see here, that is ovary always get enlarged and forms fruit. This question is asked in the examination, that which part of the flower forms fruit. So fruit is developed by fertilized ovary. In the same way, inside the ovary, ovule was present. Again, the question was asked, what happens with this ovule? So ovules are converted to seed. In this way, the embryo is present inside the ovule, which will be developing the new plant, carrying the baby plant. And the point of attachment of this seed to the wall of ovary is called as placenta. Must have seen the pea, that is green pea I'm talking about. If you are opening the pod, you will be finding a small point of attachment. I'm just trying to draw it for you at one of the place. Suppose this is the double membranous structure, and when you are opening it at this condition, a small stalk-like structure is there with which the pea seeds are connected. So this small filament-like structure is called as what? Is called as placenta, with which the ovules are attached to the wall of the ovary. Now here you can see, this is the best chart explaining the post-fertilizational changes, that ovary is converted to fruit, ovary wall is converted to fruit wall or pericarp, ovule will form seed, placenta will form stalk of the seed, outer integument of ovule will form testa or outer seed coat, inner integument will form tegmen or inner seed coat. Secondary nucleus, I told you, this is diploid. So secondary nucleus is converted to endosperm after triple fusion. Egg and synergid. Here synergid you need not to take. Remove this thing. Synergid we don't take. We take egg cell which is converted to embryo, and before embryo if you are writing here zygote, that will also be correct. And antipodals disintegrate or get dissolved. So at the last we are proceeding towards the exercise we shall be discussing in the next class. So double fertilization is what? Why it is taking place in the angiosperm? This is the important question. So in next class we will be discussing about the important questions of this chapter along with the solved exercise questions. That's it for today. Thank you.